Thank you, everyone, for joining us for the part 2 of the PSC-related cholangiocarcinoma webinar series. This part 2 is regarding translational aspects and therapy. And today we have Mitesh Borad as one of our chairs and Marcin. And our speakers are going to be Tim Greten and Sunyoung Lee will be giving our presentations today. Just want to give a couple of housekeeping notes here that the speakers have given consent to have this webinar recorded and uploaded onto the CCF website and YouTube channel for future viewing. And this was also be shared with our PSC partners, and they will be sharing it on their website as well. You, as a participant and viewer of this webinar, also agree that give our consent to attend and be recorded for any comments or questions that you may have during the recording. If you need a live certificate, certificate for live attendance, please reach out to research@cholangiocarcinoma.org and we can coordinate that with you after the meeting. And then we will have a post-webinar survey. It's a brief few questions that we would love for you to give us your input on directly after the webinar. We'll now pass it to Mitesh Borad, who will introduce our speakers.
Greetings, everyone. We're very, very excited to have two phenomenal speakers here today to discuss a very important topic in cholangiocarcinoma, and that is disease in the setting of an autoimmune disease that has been very closely linked with the disease, which is primary sclerosing cholangitis, or PSC. And we will be tackling it from two standpoints, both a basic mechanistic understanding standpoint, as well as a clinical conundrum and application standpoint. So our first speaker today is Dr. Tim Greten. Dr. Greten needs no introduction. He's the Deputy Director of the Center for Cancer Research at the National Cancer Institute and also co-directs the liver cancer program there. Dr. Greten's work has been featured in many seminal publications such as Science, Cell, and Nature. And without further ado, I'll let Dr. Greten take the fold from here. Thank you.
All right. Hello, everybody. I hope you can find my see my slides. I'm going to tell you about some research findings in PSC-related cholangiocarcinoma. I'm super excited to share this and do this. This is actually, to be honest, a topic very close to my heart because, you know, some of you may not know, but I've been working as a hepatologist for a long time, seeing patients with PSC, and I still remember, you know, how difficult it is to treat these patients and surveil these patients and make sure that they do not develop cholangiocarcinoma, and then also obviously treat them. I have no relationships with commercial entities to disclose for this presentation, which is primarily really a preclinical research presentation. Now, before we go into PSC and cholangiocarcinoma, I want to take a second to discuss immunotherapy in general in cholangiocarcinoma. And the good news is immunotherapy has arrived in cholangiocarcinoma. It has become standard of care based on these two milestone studies, the TOPAZ-1 study and Keynote 966, which are both very similar. Both used an anti-PD-1 or PD-L1 reagent in combination with the GEM-CIS backbone, and you can see the improved outcome and especially a long-term outcome with the addition of an immune checkpoint inhibitor. Now that's obviously very positive, but if we really look at this, one has to ask, is the data good enough? We have 685 patients or 1,564 patients that we needed to enroll to show very, I would say, moderate increases in median survival. So what I think, you can see here, it's 40 days or 55 days. So what I think it's fair to say is immunotherapy has clearly arrived, but the benefit remains limited. And that basically asks us for advances in basic science, because it is my belief that only this will provide the new opportunities to improve our understanding of immunological mechanisms and novel treatments, and we're conducting novel clinical trials based on those preclinical research. And this is both the case in the context of cholangiocarcinoma that's appears in the context of PSC, and to understand this better. But it's actually also something that we do in PSC, in cholangiocarcinoma in general. We actually offer patients clinical trials at the NCI, and also second opinion if this needed. And I just want to take a very brief second showing one of those studies that are based, without going into details, which are based on preclinical findings here using anti-CD40 in a mouse model in combination with another checkpoint inhibitor and chemotherapy. And you can see the outcome, which is clearly improved. And this led to a clinical trial, which we're currently doing, where we basically use the findings that we have obtained in a preclinical mouse model and evaluate this now in patients. And we enrolling currently patients and are excited to treat patients, and actually look very, the data that we have so far appears to somewhat reflect what we have seen in preclinical models. But with this, let's change now to the topic of PSC. Now, how did we get into our studies? Well, you all know that biliary cancers are a rare disease and they are difficult to find, but we do know of a patient group that has certain risk factors which are essential and which lead to cholangiocarcinoma, such as primary sclerosing cholangitis. Some of these patients also have associated inflammatory bowel disease and cirrhosis. Now, why were we so interested in this research and in these particular patients? Actually, the reason is that it was known that patients with PSC, as well as patients with inflammatory bowel disease, show specific changes in their gut microbiome. And I don't have the time now to tell you all our results from our prior studies, why we got so interested in conducting studies and trying to better understand the gut microbiome. But what was known is that patients with PSC actually have a different gut microbiome than those patients that are healthy. And you can see this here on a principal component analysis. You see patients with PSC as well as patients with PSC and different inflammatory bowel disease. And you can see they all group kind of to the right here. And in green here on the left, you have the control group, clearly showing that the gut microbiome in these two different groups is very different. If we look in more detail, you can see here at the genius level, so this is a very high level, the comparison again here, the healthy control to all the other patients with PSC and inflammatory bowel disease. And I think it's fair to say that the distribution of the different genus is very different. So with this in mind, we said, well, maybe there is something to it, and this could be used to better understand immune responses and how we can potentially treat patients with cholangiocarcinoma and potentially also with PSC and cholangiocarcinoma. So let me just tell you very quickly why we are interested in the human microbiota. I said I'm not going to show you any data, but we all know that the microbiota is basically all around us. Obviously, there are many microbial cells many, many more microbial genes than we actually have genes ourselves, and they can contribute to different biological functions. Humans have co-evolved, so these microbiota live with us. This could be bacteria, fungi, protozoa, They can be found anywhere in our body. I already mentioned that there is a lot of microbial DNA around us. It's basically an integral part of our genetic landscape. It regulates metabolic function. And even our immune system is actually dependent on the interaction with commensal flora. Now, from a mouse perspective and from an experimental perspective, we use different mouse models that had previously been described to be mouse models to develop cholangiocarcinoma. We use hydrodynamic injection of plasmids coding for oncogenes here, AKT-Notch or AKT-YAP. You can see these had previously been published, and the way this works is we use these plasmids and inject actually a very large volume of plasmid DNA into the tail vein of mice in 7 seconds. And you can see that this leads to a stasis in the liver and then a transduction of hepatocytes. The models for PSC that we used are 2 different ones. We used MDR2 knockout mice that develop here this inflammation along the bile duct, or we can perform bile duct ligation in these mice. Again, these were established models. Now, if we see now, look at the gut of mice that have PSC, you can here see the gut epithelium in a control mouse, very intact epithelium, and you can see the glands beneath. If you have now a mouse that develops PSC, in this case it's bile duct ligation, you can see that although we actually manipulate the liver, there are significant changes in the gut. You can see how the epithelium is disrupted. You can see this inflammation. You can see that markers for tight junction occludin have disappeared. We see different changes in cytokines expression throughout the gut. We can find that there is 16S RNA, which is a measure for bacteria. In the portal vein of these mice, we see that there are easily an uptake of phytidextran in these mice. And we can see even in patients with cirrhotic liver, similar to the PSC, that these bacteria can potentially appear within the liver. Now, if we look now back, if we now go back and look at the liver and look at the immune cells in more detail, and here again the data for PSC, we actually saw that there is an accumulation of so-called myeloid-derived suppressor cells, which are immunosuppressive cells that we can find here in the liver of mice with PSC. We can subdivide and look at these MDSCs in more detail, and we find that the majority of these cells are so-called PMN MDSCs or polymorphonuclear MDSCs. The same applies to the second model that we have in our mouse model. And the interesting thing is, if we treat mice now with neomycin to deplete bacteria, this accumulation is reversed. It no longer appears, clearly indicating that bacteria in the gut are needed to induce this accumulation of these immunosuppressive cells in the context of PSC. Now, in order to show this in more detail and to prove this further, we perform so-called FMT studies. These are transplant studies where we use fecal samples from mice that have been kept on regular conditions and were treated with 2 different antibiotics, vancomycin or neomycin, so that we deplete either gram-positive or gram-negative bacteria. We transfer these into germ-free mice, which are completely bacteria-free, and then we look after 2 weeks at the cells in the liver, and we see that those mice that have received the stool samples from mice treated with vancomycin, so these are all the gram-positive are eliminated, so the gram-negative are left. And basically, if we transfer these gram-negative bacteria, you can see that you find again this accumulation of these polymorphonuclear MDSCs. So we looked at these MDSCs in more detail by flow cytometry. And we noticed this accumulation of polymorphonuclear MDSCs, which actually express a chemokine receptor CXCR2. And CXCR2 binds to CXCL1, so we got interested in looking at CXCL1 in the liver. Indeed, we find increased levels of CXCL1 in the context of PSC, but actually, interestingly, also in the context of colitis. The CXCL1 is mainly produced by hepatocytes. And if we block either CXCL1 using an antibody or using a small molecule, we can actually inhibit the accumulation of these cells in the liver. And this, again, is dependent on the expression of CXCL1 in the liver and the gram-negative bacteria, because what we show in this experiment down here on the right, the gram-negative bacteria bind to the TLR4 receptor on hepatocytes. And if we actually use a TLR4 knockout in the liver, as shown here, we no longer find this accumulation of myeloid-derived suppressor cells. So let me summarize what I've shown you so far. So in the context of liver cirrhosis, we develop a leaky gut. Gram-negative bacteria, or LPS, go through the portal vein into hepatocytes. The hepatocytes start to produce through TLR4 CXCL1, and this leads to the accumulation of CXCR2 positive polymorphonuclear MDSCs. And again, this is dependent on gram-negative bacteria. Now, at this point, I was able to show you the context of bacteria and these immunosuppressive cells, but we haven't really talked about cancer yet. So the next step was to combine those models that I have just shown you with these models that we have in the lab that I had explained initially and described initially, where mice will develop cholangiocarcinoma by hydrodynamic injection. And you can see, indeed, if we perform bile duct ligation to induce PSC, there are more and larger tumors in these mice, both after bile duct ligation as well as in the MDR2 knockout mice. We can also mimic this in a mouse model where we induce colitis. And again, we looked at our MDSCs, and you can see that these cells are indeed increased. We can manipulate this, we can manipulate the tumors if we eliminate these cells. So if we use an antibody to eliminate myeloid-derived suppressor cells, we find fewer cells. If we overexpress CXCL1 so that we get more MDSCs, we find more tumors. If we block CXCL1 with an antibody, again, we find fewer tumors. I've already explained earlier to you the experiment where we use an FMT. And again, here, if we actually deplete gram-negative bacteria in this FMT study, we also find fewer tumors in this model. The mechanism, again, depends on the toll-like receptor 4 expression on hepatocytes and is dependent on CXCL4. First, you see the reduction if we knock out the TLR4 receptor on hepatocytes, and then we reverse this if we can overexpress CXCL1 in this system. And downstream of this myeloid-derived suppressor cells, we believe that the cells that are responsible here for tumor rejection are mainly NK cells, because if we deplete this, we no longer find a difference. So let me summarize this. So what I'm trying to explain to you that here in this context of PSC, there's actually a very interesting phenomenon. We have the liver that secretes, that communicates with the intestine in the microbiota. Now, if there is a reason to develop fibrosis and cirrhosis, such as in the context of PSC, what happens is that there are changes in bile acids and there is dysbiosis. So dysbiosis is the change of the microbiome. And this change of the microbiome leads to a leaky gut and to inflammation in the gut. I've shown you those images from those mice where we induced PSC, and then although the whole scenario was actually the manipulation was happening in the liver, we saw actually changes in the gut. Now, with this leaky gut, bacteria, and here the gram-negative bacteria are important, can actually migrate through the gut epithelium. They go through the portal vein into the liver, and in the liver, these gram-negative bacteria are being go to TLR4 positive hepatocytes. The hepatocytes start expression of CXCL1. CXCL1 in turn leads to an accumulation of CXCR2 positive granulocytic MDSCs. And these MDSCs are immunosuppressive and can basically block the function of NK cells. And in the absence of NK cells, we have enhanced tumor growth. So I believe what I've tried to show you here is a mouse model of PSC-related cholangiocarcinoma. This model helps us better understand the disease per se. In this particular study, we took a specific look at the gut microbiome and how actually this crosstalk between the liver and the gut and changes in the gut and the gut microbiome then in turn can lead to an immunosuppressive microenvironment and thereby promoting tumor growth. And with this, I'd like to thank you. I'd like to thank the organizers for allowing me to talk, and I'm happy to take any questions.
Thank you, Tim. That was fantastic. A lot of questions come to mind based on what you showed, especially for patients who have PSC and maybe haven't yet developed cancer. Obviously, not everybody does. So based on the work you showed with the microbiota, would, say, fecal transplants be a strategy, or would changing the microbiome profile of patients who have unfavorable to the more favorable group that you showed be an approach?
I think that's a very interesting and important question. And we know that PSC is actually obviously a very difficult disease because it is often also associated with inflammatory bowel disease, as I just mentioned. And obviously here, the gut microbiome may even play a much bigger role. And there are numerous attempts right now, to manipulate the gut microbiome. And one of those attempts may actually lead to changes in this scenario that I've just shown you. So I think from a theoretical point of view, this is a very interesting approach. I know there are many trials ongoing in the context of inflammatory bowel disease. I know there are trials ongoing in the context of immune checkpoint inhibitor therapy and cancer. I know there is a lot of interest in studying the gut microbiome in PSC. I'm not aware yet of trials specifically in this scenario, but I think this is certainly something that that could come into your mind.
And what about the role of antibiotics as a therapy, or adjunct therapy at least, in management of patients both before developing cancer and after?
Yeah, that's a long question. That's been going on for quite some time, and and I'm always a little bit cautious because if you have a medical provider that prescribes antibiotics, there is a reason for that, especially in the context of PSC. These patients develop cholangitis and a lot of times require antibiotic treatment. Yes, I do acknowledge that the antibiotic is going to change your gut microbiome, and it may potentially change it not in a way favorable that you think immediately, but this may be a long-term effect. And I think, it's something that to weigh in. And I would be, especially if there is a patient, listening to this, I would be very careful, to make too strong of a statement based on preclinical studies right now, to be careful with an antibiotic treatment.
And these microbiota that are there, is it their presence in terms of antigens that will be presented by the immune system that causes the inflammation, or do they themselves secrete substances that are inflammatory?
Well, this is an interesting question, and there's various mechanisms how they work. the one mechanism that we know and that we have shown here, and that I think is also probably happening in a lot of patients, is through toll-like receptors. So toll-like receptors basically, are a way of the human immune system to early recognize foreign invaders. And they basically cause responses of the innate immune system and not an antigen-specific response to begin with. The antigen-specific response usually takes some longer time, but yes, we do know that you can actually also develop antigen-specific responses to certain bacteria, and there may actually even be a cross recognition between certain bacteria in tumors. And if this was not complicated enough, we know now, using more sophisticated techniques than the ones we have used, that some of these bacteria not only migrate to the liver in the portal vein and then disappear, we actually find intratumoral bacteria. And there are a number of studies also in the liver, where investigators have found that within the tumor cell, there are actually specific bacteria. Now, we don't know exactly how these bacteria get in there. And especially coming back to our today's topic, PSC, it's not necessarily clear if they come through the portal vein or through the bile, because we know that obviously, the bile is, that the bile duct is another connection, which is actually way shorter than the portal vein. And especially in the context of a patient that has biliary obstruction, where the bile doesn't flow that fast, I think it's very reasonable to believe that bacterial colonization may actually also occur through the bile.
Okay, thank you. And are there questions from the audience at this time for Dr. Greten? And yeah, just feel free to come off your camera and ask if you have something right now. Okay.
Hi.
Hello. Yep.
Hello.
Yeah, and please
Yeah.
introduce yourself briefly and then go ahead and ask the question.
Hi, I'm Jovan, a postdoc in University of Copenhagen. I have a question about the selection of CSCL11, because like when doing omic data or whatever research, there's always lots of cytokine, lots of CCL, lots of CSCL appears. And is that CSCL11 ran the top when you doing kind of screen, or maybe I missed how you select CSCL11 to go further at last.
Okay, so this is an interesting question. So let me just make sure. We actually found CXCL, we actually talk about CXCL1, not CXCL11. And this was not found through a research, through a screen, but this was an educated guess, because if you know the chemokine receptor, so in this case, we made the observation that the MDSCs accumulate, and we found that these accumulated MDSCs express CXCR2, which is a receptor for CXCL1. So that basically triggered our interest in this particular chemokine.
Okay, well, thank you for that question. In the interest of time, we'll get started with our second part here, and Dr. Sunyoung Lee will be presenting this part. Dr. Lee has been a very prolific investigator in the field of cholangiocarcinoma. He's in the Department of Gastrointestinal Medical Oncology at MD Anderson. And without further ado, turn the floor over to Dr. Lee.
Thank you so much. Let me share my screen. Perfect. Thank you so much. My name is Sunyoung Lee. I'm one of the GI medical oncologists working at MD Anderson Cancer Center in Houston, Texas. So I'm going to discuss PSC-related cholangiocarcinoma, mainly focused on the clinical outcomes data and DNA alterations and gene expression profile. Oh, perfect. Okay. As we all know, PSC, primary sclerosing cholangitis, is a strong risk factor for cholangiocarcinoma. We have analyzed 57 patients with PSC and cholangiocarcinoma, it's a biliary tract cancer, treated at MD Anderson Cancer Center and Princess Margaret Cancer Center in Toronto, Canada. Out of 57 patients, 43.9% of patients had intrahepatic cholangiocarcinoma, about 30% hilar, 19% distal cholangiocarcinoma, and gallbladder cancer was 5.3%. Radiographically, 44% of patients had a periductal infiltrating tumor, and 24.6% here, 14 patients had a mass-forming tumors. 57% of patients had hypovascular tumors. 7.3% peripherally enhancing tumors, and 35.7% had hypervascular tumors. 29.8% of patients only had PSC. 42% PSC and ulcerative colitis at the same time. 16% of patients had PSC and Crohn's disease. 12.3% had PSC and other autoimmune conditions, including PBC, autoimmune hepatitis, and celiac disease. In terms of median overall survival, when median OS was compared in patients having PSC cholangio and non-PSC cholangio is a control group, there was no difference in terms of median OS. The P was 0.6 and 8. Surgical interventions, local regional therapy showed survival benefit. For example, 33% of patients had surgery, 1.7% of patients had a liver transplant, and the recurrence-free survival was 11.8 months, and the median OS was median overall survival was 52.2 months. 24% of patients received radiation treatment, 1.7% Y90, 1.7% ablations, and event-free survival was 8.3 months, and median OS was 19.2 months. Patients who only received medications had a median OS of 16.2 months. So the bottom line is here, we need to maximize surgical and the local regional therapy for patients having PSC and the cholangiocarcinoma. So in terms of systemic treatment, these are, the table shows chemotherapy and the systemic treatment options. So first line treatment, obviously gemcitabine, cisplatin, gemcitabine paclitaxel, gemcitabine, nab-paclitaxel, gem-K, FOLFIRINOX, FOLFOX, and second line, similar medications. And in terms of median PFS, first line PFS was 5.9 months, and the second line PFS was about 4 months. Median PFS of gemcitabine-related regimen, gemcitabine, cisplatin, gemcitabine, nab-paclitaxel, and other medications, versus others was 5.9 months versus 4.6 months, but P was 0.9, so there was no difference statistically. Median PFS of 5-FU-based chemotherapy and others, there was no difference. In terms of immunotherapy, out of 57 patients, 14 patients received immunotherapy, either anti-PD-1 and anti-PD-L1. 6 patients received immunotherapy in the first-line setting. 8 patients received immunotherapy in the second-line setting. When patients received immunotherapy in the first-line setting, median PFS was 13.3 months versus 4.7 months, and the P was 0.06. Second-line PFS, when immunotherapy was given in the second-line setting, was 7.4 months versus 3.9 months, compared to patients who didn't receive immunotherapy. But P was 0.47. It was not statistically significant. Response rate was 29% from immunotherapy, and 13% without immunotherapy, and P was 0.248. And disease control rate was 86% with immunotherapy versus 43% without immunotherapy, and P was 0.006. One patient who received gemcitabine, cisplatin, and durvalumab had grade 3 colitis, but other patients didn't have any immunotherapy-related side effects or immunotherapy-related PSC, UC, or Crohn's flare. And this is a table showing 14 patients. For example, when we look at the patient number 6, a patient received gemcitabine, cisplatin, and durvalumab, and the patient had a complete response, and the patient has been on durvalumab maintenance therapy. Patient number 7, who had PSC and also UC, and the patient had a grade 3 colitis when durvalumab was given combined with gemcitabine and cisplatin. But the grade 3 colitis resolved spontaneously when we stopped immunotherapy. The last column that I labeled in purple color, so many patients received medications to control and prevent PSC or UC or Crohn's flare. Many patients received ursodiol, mesalamine, and other medications to control these medical conditions. This is the data on the genomic analysis. As we all know, cholangiocarcinoma have well-known DNA alterations. For example, KRAS mutations, MTAP loss, MDM2 amplification, BRAF, ERBB2 amplification, FGFR2 alterations, including FGFR2 fusions and rearrangements, IDH mutations, and others. In terms of DNA alterations in patients having PSC and cholangiocarcinoma, p53 mutation and KRAS mutations are the most common mutations. At the same time, many patients had both T53 mutations and KRAS mutations together. T53 mutations or KRAS mutation alone or combined together were related to worst OS, overall survival, and there was a trend toward worse PFS to systemic treatment. In terms of KRAS mutations, 9 patients had KRAS G12D as in David, 2 patients had C, G12C, G12A, G12R, and then other mutations. There were no FGFR2 fusions or rearrangement. There were no patients had IDH mutation or ERBB2 amplification. I recently treated one patient who didn't have PSC but only had ulcerative colitis, and patient had FGFR2 fusion. But from this this data, current data, especially patients having PSC, we didn't see any patients having FGFR2 fusion here. One thing to be noted is PSC and cholangiocarcinoma tumors had a quite increased tumor mutational burden. For example, 9.2 patients who had PSC and cholangiocarcinoma had a 9.2-fold higher tumor mutational burden compared to patients having cholangiocarcinoma but no PSC. 6 patients out of 16 patients having the data here in PSC cholangiocarcinoma had a TMB greater than 10, but 0 patients out of 44 patients in non-PSC cholangiocarcinoma had the TMB greater than 10. 2 patients had MSI tumors in PSC tumors. And in terms of pathogenic pathways, there were similar distributions of DNA mutations here, including P53 pathway, cell cycle pathway, RAS and RAF pathway, epigenetics, and TGF-beta, and others. In terms of whole transcriptome analysis, there was no difference in terms of messenger RNA expression profile of genes related to immune checkpoint molecules here, PD-L1, PD-L2, PD-1, and CTLA-4. And so the figure B on the right side shows that even in patients having PSC and cholangiocarcinoma, some patients had immune hot a profile, but other patients had immune cold profile. Immune response signatures here, immune system being hot or cold, were independent of tumor mutational burden. And then, like I mentioned, there were 2 patients who had MSI high tumors, and that they were immune cold here. And that the PSC and UC or Crohn's cases with immune inflamed like phenotypes, all intrahepatic cholangiocarcinoma cases. Like I mentioned, there were distinct group of patients who had immune inflamed features, and there are patients whose the tumor microenvironment was not really hot. High TMB tumors responded better to immunotherapy, or they had a longer disease control rate to immunotherapy. But there was a patient with TMB of 0 and had a prolonged response. So going back to the same table, so once again, when we go back to the same patient, patient number 6, who received gemcitabine, cisplatin, and durvalumab, patient's TMB was 0, but patient had a complete response. And the patient has been on the treatment for a long period of time on durvalumab maintenance at this point. Patient number 12, patient had a tumor mutational burden of 186.3, and the patient had a prolonged but stable disease, not a PR or not CR, but a prolonged response, a stable disease. Patient number 14, who had PSC and UC, who has received gemcitabine, cisplatin, and durvalumab, and the tumor mutational burden was 48.8, and the patient has been on the same regimen for a long period of time. Immunotherapy is considered quite safe, and actually safer than we expected, and there was no major concern about autoimmune flare. So we get a decent number of patients having PSC, UC, or Crohn's, and autoimmune hepatitis, and we are all concerned about immunotherapy-related flare. So a patient received gemcitabine cisplatin only, and then coming to MD Anderson for a second opinion. We always share this data. Immunotherapy is related to longer PFS and a better disease control rate, with a trend towards better OS and response rate when patients receive immunotherapy. There were no patients having IDH1 mutation or FGFR2 fusion, and the KRAS mutation and or TP53 mutations were related to a worse clinical outcome. We all know that TMB is generally low in biliary tract cancer, but there were a lot of patients whose tumor mutational burden was greater than 10 in PSC cholangiocarcinoma. We have not accurately identified good, perfect genomics marker to predict a response to immunotherapy. And then we are working on analyzing patients having cholangiocarcinoma with no PSC, but other autoimmune diseases, including IBD and the celiac disease. Thank you so much for this great opportunity. So the study was performed with my patient, Mr. Hausman family, and also Athan's family, and is also the grant from MD Anderson Cancer Center. Thank you.
Okay, well, thank you, Sunyoung. That was fantastic and a lot of relevant data for patients in particular. The key one is obviously the safety of giving these checkpoint inhibitors with gemcitabine and cisplatin in advanced disease patients. So thank you for showing that. Was there any difference at all in patients who had some immunosuppression going on with some agents versus those that may not have sufficient immunosuppression?
Yeah, so I think that's a great question. So we actually, the data is under review in a journal, and this was the question from the reviewers. Yes, so the study shows that is we basically had a control group having cholangiocarcinoma but no PSC versus PSC and cholangiocarcinoma, and then as I showed you in the patient table. The last column. So we had a good control. When we compared the outcomes data in patients having patients who received mesalamine, ursodiol, and other medications, so we had a good control group, and there was actually no difference in terms of clinical outcomes data, especially response to immunotherapy.
Wonderful. Yeah, we have a few other questions from the audience here. So, Green, okay, you want to?
Hi, thank you, Mitesh. Really nice presentation. I wanted to ask you, it was kind of surprising to see 14% of those included were mass-forming tumors. Is there any chance that they were mixed tumors? And have you looked at those specifically to see if they harbor any particular alterations? Because if I'm right, you're saying there were no IDH1 mutations or FGFR2 fusions found. Is that correct?
Yeah, that's correct. Yeah. So I think in terms of the mixed tumors, so you're talking about the combined ACC and cholangiocarcinoma?
No. I'm talking about mass forming versus periductal and the mixed growth patterns that you can see, because mass forming tumors coming from PSC cholangio is I would have thought rare because they tend to be large up tumors. And I'm just wondering if you looked at those specific mass forming tumors that you had to see whether they harbored any particular alterations like absence of KRAS or absence of P53. They would be particularly interesting.
Yeah, so, I don't think we have specifically analyzed that to say like was there any difference in terms of DNA alterations, KRAS, with and without KRAS or T53 mutations in mass forming tumors or not. So we don't have data, but I'm happy to go back to our radiologist who reviewed the scan and then compared the clinical outcomes data, also the DNA alterations. There may be a potential questions from the reviewer. So yeah.
Yep.
We'll turn it over to Tim for the next question.
Yeah, that's a very interesting data. And obviously what is intriguing is, we're dealing with an autoimmune disease, but the majority of the patients actually don't really receive immunosuppressive treatment because, because of PSC. So I was wondering, you still had a very high, good number of patients with IBD. So I understand this was not part of a clinical trial and it's a retrospective study, which is all fine. But did you actually happen to treat any patients that had active inflammatory bowel disease? And were there any patients that, had maybe recently received steroids or something? Or did you exclude them on purpose? Because, I would assume that these patients are at much higher risk for a flare.
Yeah, thank you so much for the great question. Yes, so we do have some patients who have been taking steroid, like a 5 milligrams of prednisone. Some patients were really actually taking azathioprine and other medications that are actually suppressing the immune system. So yeah, I mean, a lot of patients had a quite well-controlled and stable UC or Crohn's, but there were some patients who were actually, getting medications to suppress the immune system, and then we didn't really see a lot of flare here.
Interesting.
Okay. Juan, do you have a question?
Yes, thank you. I was going to ask the same question as Tim, but I'll just pivot to a different question. In your experience, and maybe looking at the literature, do you find that patients with PSC-related cholangiocarcinoma are more likely to be diagnosed at an earlier stage because they're being monitored, surveyed, versus de novo?
Yeah, I think that is probably true. Although, so when I go back to my slide. Sorry, for some reason I'm not able to.
Yep.
Yeah. I don't think I mentioned it here. Yeah. So there were in terms of staging, stage 1, 2, 3, and 4. There are a lot of patients who had stage 4 here, 36%. And then there were this number of patients who had stage 2 disease. And that's why, like I mentioned, there was about 33% of patients, did undergo surgery or liver transplant here, right? I mean, but this is a retrospective data analysis, and this is not a clinical trial. So it is, in terms of incidence and anything, there are a small number of literature and the journal articles showing that because patients having PSC are getting a very frequent follow-up and scans, and even like ERCP and ultrasound and MRIs. And then some studies did show that these screening processes can potentially decrease the cholangiocarcinoma, I want to say the bad clinical outcome here. So, and then also there are a few journal articles showing that PSC-related cholangiocarcinoma is diagnosed earlier than other patients having no PSC cholangiocarcinoma. So I think that's a great point. But in terms of here, our data analysis, I put a control group here, and then we had a good control here. So similar cancer stage and also other clinical outcome data here.
Thank you.
Okay, now there's a comment in the chat regarding the Topaz and Keynote 966 studies not having these patients. So this real world retrospective data is going to be critical to building confidence for use of these agents also in this patient population.
Yeah, I think that's a great point. Yes, when we run clinical trials like a Topaz 1 trial and other trials, almost all cases we explore patients having autoimmune disease when patients receive immunotherapy on the trial. Yeah, it's true.
Okay. All right. Well, any other questions from the audience? All right. If not, I want to thank both of the speakers, Dr. Greten and Dr. Lee, for those fantastic presentations, and for the audience for that very interactive session. I'll turn it back over to Ash Patel for some closing remarks.
Once again, I want to thank everyone for your participation in this webinar series. Our next webinar is still to determine. We may expect to have one sometime late summer, early fall, August, September timeframe. So please look out for more information about the next webinar on our website. And then you can also be receiving some emails if you're an ICRN member. If you are not an ICRN member currently, I encourage you to use the barcode you see on the screen to join. We have reconfigured our committees, and we would love for you to join and participate actively in one of our committees and network with some of the great scientists and research across the globe. Lastly, we put a link in the chat feature, but also here, if you could take a moment and fill out the post-webinar survey. Your input is very informative for us to improve these for any future sessions that we do. If you need a live attendance certificate, feel free to reach out to us at research@cholangiocarcinoma.org, and we could coordinate that with you. Once again, thank you for your participation. Thank you to the speakers, and thank you to the moderators for doing an amazing job. Thank you.
Take care.
Bye-bye.